morning. Good morning, dear friends. I'm very happy that you are here today. To our university, we have a great honor with lecture. We invite the Professor Lawrence DuBasке, the clinical professor of medicine in the George Washington University School of Medicine and Health Service, director of the Immunology Research Institute of New England. Professor Lawrence, the floor is yours.
Thank you so much. It's an honor to be here. I want to thank the rector for allowing me to speak with all of you. I want to thank my good friend, Professor Helena Koval, who I've known for probably 10 years and have collaborated with for arranging this tour. And I want to thank all of you for taking some time to be here with me today. As you heard, I serve at George Washington University. I've been there since 2010. I've been a clinical professor of medicine in the Department of Allergy and Immunology. Prior to that, though, I served 30 years on the faculty at Harvard Medical School in Boston. And I was the Allergy Fellowship Training Program Director at Harvard Medical School, Brigham and Women's Hospital. Before that, I went to medical school at Northwestern, and you'll hear a little bit more about that in my lecture. I've been in this field of allergy since 1976. So I've been doing this for 50 years. And after 50 years, you kind of get a different perspective on things. I started a nonprofit foundation, Immunology Research Institute in New England, in 1989, which was designed to help physician scientists from Eastern Europe, essentially. And I'll explain why in a few moments. Since 1989, I've been coming on a regular basis to various parts of Eastern Europe. It actually began in 1987 when I first visited Poland during martial law, when Wojciech Jaruzelski was running the show there. I first came to Ukraine in 1993, shortly after independence of Ukraine. And I've been coming here since that time. So I've watched your country develop, I've watched wonderful things, and I've seen some of the horrors which have happened recently. And I want to first say I am honored to have all of you here, but I really salute the strength, the courage, the defiance of the Ukrainian people. I think it's awe-inspiring, and thank you for allowing me to be here among you. So, in the U.S., there's this thing you do, you put a disclosure up. It means who you've worked with and what you've done. And basically, I work with all the pharmaceutical companies that are involved with biologics. I've also worked for all the companies that are involved with allergen immunotherapy. Most of what I'm going to talk about today is work with one company called Allergy Therapeutics, which is a company out of the UK, whose work has interestingly sort of paralleled a lot of my interest in my career. Well, here are my parents. You have to, in the beginning, Stanislaw Szyborski and Josefa Shanowska are my parents. Here they are in 2016. They both lived until 97 years of age. My father's father was from the Krakow region, my mother from the Galicia region of Ukraine. Here's her mother. So this is truly in the beginning. Katarzyna Saganik Shanowska immigrated from Sanok, which is now in Poland, but was a Ukrainian region of Poland, and she came to the United States around 1905. In 1907, she married Josef Schanovsky, and here's their wedding photo from New York City. Josef was from Uknev, which is in Lviv Oblast now, and a small village north of Ravaruska, if you know the territory. So they met in New York City. They got married in New York City, but they both were from Galicia. And they didn't waste much time. Over the next 10 years, they had 8 children. And my mother is actually pictured on her mother's lap. And she was a beautiful woman, and even more amazingly, a beautiful woman after having 8 children in 9 years. So amazing story, and a very strong woman. Now, my grandfather's sister is the mother of this woman. Her name is Stanislava Bunkovska-Zemnievich, and she also came from Uknev, since her mother and my grandfather were brother and sister in the Lviv region. She lived to 101 years of age, and is a testimony to human strength. She was still lecturing as a teacher into her 90s and writing for a local newspaper in this town of Dukla, which is on the border, but on the Polish side of the border. And here she is with her son Zizek in 2012. Zizek still lives in this area. Here he is. He's now 78 years of age. Here he's pictured in 2016. As I say, I came to Ukraine based on my familial interest in this part of the world, quite frankly. And in the early 1990s, 1993, I came to Kiev. First time I went to the Tuberculosis Institute. I met Professor Nikolai Kovalenko. One of his students, Alex Markov, became a lifelong friend. Alex's brother, Dmitry Markov, was Ukrainian ambassador to the Netherlands for a while. And I got to know through Markov, Ambassador Sherbak, Yuri Sherbak, who was a physician, was ambassador to the United States, and his daughter, Tatiana Gutz, was also a physician and worked at my laboratory at Harvard University for a while in the 1990s. So this is all, became like a large family. I was there in 1987 when the Ukrainian Allergy Immunology Society was founded, August 5, 1987, and I got to meet a lot of people, and I'll just show you a few of them. I helped sponsor many scientists from Ukraine, and when things were friendly, Belarus and Russia, to meetings internationally. And here is the European Academy of Allergy and Clinical Immunology. This is Istanbul, Volodymyr Beloglazov, who is from Simferopol, and Igor Kaidashev, who is now president of UTI from Poltava. Here he is in Milan, 2013. I'd helped again sponsoring these posters that are being displayed were posters I helped them develop and printed and brought to these meetings. Here's Valentina Chopyak with Igor in Milan, 2013. And here they are again, is where I'm going next, Vika Rodenkova. I'm going to Vinica next, along with Igor Kaidashev. And here they are at what we had Irene Symposium, Immunology Research Institute of New England Symposia at the European Academy of Allergy in 2013. My good friend, Professor Yuri Brusyuk, was in Kiev at the time of the war. 2022 Prague, Professor Galina Koval and Professor are finally with a flag that they brought, and this is in Philadelphia, and I love this photo at the American Academy of Allergy meeting. So now on to my lecture. I've been involved with allergy and immunotherapy since 1976 when I was a student at Northwestern University in Chicago. And issues with allergy shots or allergen immunotherapy include safety, because every year people die from anaphylaxis from allergen immunotherapy. And they're usually being treated for respiratory disease like rhinitis, and nobody dies from rhinitis. So there's really no excuse for having somebody die from a treatment for rhinitis. There's been a lot of literature worldwide recently that adherence to allergy shots, allergen immunotherapy, is very poor. And efficacy, while good, could be a bit better. And long-term efficacy requires 3 years of treatment. 2 years doesn't give a long-term benefit. 1 year obviously doesn't. And in a publication I had with, again, with my institute, collaborated with Mitja Koznik, who's in Slovenia. We did a study showing that 5 years of venom immunotherapy gave a 10-year benefit. So we think probably even longer is better. But the problem is less than 15% of people achieve even 3 years of treatment, and you need that to get a long-term benefit. So possible solutions. Let's make it safer. And that's where Northwestern University came in. I worked for Professor Roy Patterson, who espoused modified allergens, allergens that you chemically change, and I'll show you in a minute how you do that, to make them safer. Trying to get a shorter course so people would really do it. But what you had to do was something that my friend Professor Patterson did not have access to, and that is you have to give an immune boost, which is called an adjuvant, in order to get the best response. And we'll talk about the adjuvant that I'm involved with which is monophospholipid A, MPL, which is an endotoxin-like substance derived from Salmonella minnesota. So allergoids. Allergoids are modified allergens designed to be safer. And the typical way of making an allergoid is to combine the allergen with glutaraldehyde polymerization, where you cross-link epsilon amino acids on the proteins. And what ends up happening is you get something that kind of looks like a ball of spaghetti. And it causes the allergen to lose the epitopes, the part of the allergen that binds to immunoglobulin E, but maintain shorter epitopes, which are T cell epitopes, that allow you to use it as an immune modulator. Again, Roy Patterson at Northwestern University pioneered this in the 1970s, beginning with polymerized ragweed. And then my colleague, Alexander Babakin, who was in Moscow at the time and went to work for a while with Bencard in the United Kingdom, took it further, developing a number of polymerization techniques and adjuvants. Now, just so you understand, Babakin and I first met in 1989 in the United States in a meeting. And we began what was to be a 30-year friendship until he passed away. He was in my house staying for 6 months at a time, working in my laboratory in the 1990s. He was a very creative man developing polymerized allergens, but his work is rather obscure except for some of the things that he and I published together. Most of his work is in Russian language, in Soviet and then Russian literature, but he did great work. But the end result with his approach and Patterson's approach was that you got safety, which was enhanced, and you could use less injections. So you begin with a raw material. You extract the allergen from the raw material. It's grass pollen, ragweed pollen, and then you add glutaraldehyde to form an allergoid, the polymerization. And what happens is shown here. On the right is skin test reactivity. On the left is reactivity against immunoglobulin E and immunoglobulin G. You maintain immunoglobulin G reactivity, which are some of your T cell epitopes, but you lose immunoglobulin E reactivity, which means your skin test essentially gets wiped out. You're not reacting to the allergen if you do a skin test with this modified allergen. And this is an allergoid and should be safer. Here's Professor Patterson. Again, I first met him in 1976 in Chicago. He was a very interesting guy. He played a musical instrument called a recorder. It's sort of like a long flute. They kept it in his office. His family was from Finland. He died a tragic death about 20 years after I met him from a brain tumor. But it was one of the real pioneers in the field of allergen immunotherapy. And he did a study with ragweed, shown here and he did it together with Albert Scheffer, who's the senior author on this piece. Albert Scheffer later became my mentor at Harvard University, and I had the pleasure of working with him for 30 years, the entire time I was at Harvard. But Albert Scheffer and Roy Patterson were good friends, and they published this work showing that essentially anaphylaxis was wiped out, and they could get an effect in between 11 and 13 injections, an effect of desensitization. So the idea was here a 15 injection schedule, no systemic reactions, safety was enhanced, and the cumulative number of patients were almost 300 people, 435 injections. You didn't need a lot of injections to get the benefit. And here's the result of Patterson's work. The people who got the modified allergen had less symptoms in the first season than the people who got placebo shots, shown as the top line. The bottom line is the modified allergen. So it looked to be effective. They then finessed it a bit, and they went to grass, and they found they could do it in 13 injections, and they had no systemic reactions. Now, unfortunately, this was a project done solely by the university. He had no pharmaceutical money, and that really was what stymied him in terms of bringing it to the marketplace. But his work did not go unnoticed. The fact that you could have a short schedule, get to a very large amount of allergen quickly, and have no systemic reactions, that idea was picked up by a company in the UK called Bencard, and they saw this and they decided to run with it. Meanwhile, in Moscow, this man, my friend Alex Babakin, also was reading this literature, and he began at the Russian Federal Institute to develop all sorts of polymerization techniques. And he showed in animal models that these techniques could give you polymers or monomers that lost immunoglobulin E reactivity. But the problem was that they lost some immunogenicity when you did this. So they were still able to give some level of desensitization. It just wasn't sufficient. And he came up with the idea, well, if you add an adjuvant, an immune stimulator, that probably you could get a better response. So modified allergens I guess I just hit some. Modified allergens with immunoadjuvants were then developed by Bencard. And Bencard looked at microcrystalline tyrosine and monophospholipid A and putting them together to form an adjuvant. And more recently, this company, Bencard, evolved into Allergy Therapeutics, has looked at virus-like particles that have two adjuvants, tetanus toxoid and E. coli RNA, to produce an adjuvant system. The virus-like particle gives a similar situation to a modified allergen, in that the virus-like particle, you can bind your allergen, your components on it in such a way that it won't cross immunoglobulin E molecules and it won't induce anaphylaxis. So this is how you bind the particles. So monophospholipid A was developed by a company in Seattle, Washington called Corixa, that was later taken over by GSK, GlaxoSmithKline. It's a TLR4 agonist, toll-like receptor 4 agonist. It's a very strong Th1 response, and it's in the major vaccines now for respiratory syncytial virus and for herpes zoster virus. Both are produced by GSK. And I was in Poland lecturing at the Polish Allergy Society meeting last week on Thursday, and I saw GSK had their exhibits with Arexvy and Zostrix. These are the two vaccines that are now becoming worldwide that have MPL as important component. Steve Durham did mechanistic studies. Steve is from the UK, on the combination of MPL and modified allergen, and he showed that you got a very strong Th1 response. It was monocyte dependent, and it led to a reduction in interleukin-5, an increase in interferon gamma on these monocyte cultures that were cultured with the allergen, and no change in interleukin-10, which is normally thought as an immunoregulatory cytokine, but it was solely based on giving a big Th1 boost. And what he showed was that you had an interleukin-12 dependent response. If you took interleukin-12 out of the system, you lost the response. So monophospholipid A drives interleukin-12 production in a way when combined with an allergen that is allergen specific. And it causes a reduction in interleukin-5 production and an increase in interferon gamma production, and it's monocyte dependent. As we now know, T cell dependent. So GSK at the same time combined MPL with something called ASO1, which is an adjuvant system containing a saponin. Saponins came from tree sap. That's how the name saponin. And they are very strong promoters of Th1 and responses involving interleukin-12. So here you see interleukin-12 monocyte dependent. It's in the RSV vaccine and it's in the herpes zoster vaccine. And what's amazing, the RSV vaccine has been shown one injection containing this monophospholipid A will give you 3 years of protection against respiratory syncytial virus. 2 injections of the herpes zoster vaccine will give you, it appears now, 12 years of protection, just 2 injections against herpes zoster. And they did a lot of work, I'm just going to quickly show it, showing that there was a synergy between MPL and the saponin, and that you got an impact again on macrophages, interleukin-12 dependent, you got an inflammasome reaction, and this inflammatory response induced by the MPL promotes reaction with the antigen you give. In this case, herpes zoster, it could be RSV, or in our case, the allergen that's modified. The RSV work was published rather recently, showing a big effect for 2 years. They now show 3 years, and a great immunologic effect from this vaccine. Also, what they showed is that CD4 T cells activation markers persisted at least for a year after the immunization, and that it was safe and effective for 2 seasons. Now they published 3 seasons, and they've shown it is safe compared to placebo vaccine, and really had only local adverse effects, a bit of swelling in the arm, some redness due to the effect of the MPL. But they had a prolonged immune response with zoster, even a more prolonged immune response. And here's data with zoster showing that the antibodies that are produced will be persistent, and here it's for 8 years after immunization, and T cell responses persist for 8 years after just 2 injections against zoster. And long-term, now they've shown evidence of 10 years of protective effect, and they show here year by year, the overall efficacy remains in around 80%, going all the way down to 10 years after immunization with this approach, and immunologic effects that persist 10 years in terms of T cell effects and antibody production. So, getting back to my field of allergy, using this approach, MPL, these 2 men, Alan Wheeler on the top, Stephen Warnetsky on the bottom, came up with a system that was marketed by Allergy Therapeutics and before Bencard, called Pollenex Quattro. And this involves a modified allergen with MCT, which is microcrystalline tyrosine. It was actually discovered. This is another adjuvant, discovered in the 1950s by this man, Ethan Allen Brown. He is one of the founders of the American College of Allergy. He came from the UK. He was a professor in London, came to Boston, brought his interesting scientific expertise to the United States. and practiced allergy until his death. He had a big clinic, and when I began as an allergy fellow in 1981, I was fortunate enough to inherit some of his patients. But at any rate, he discovered microcrystalline tyrosine, and it's a short-term depot. It gets absorbed in about 3 days. Microcrystalline tyrosine has not been shown to have any toxicity, but it reacts with something called chi forces. These are electrical forces. It will hold the allergen and hold your adjuvant together, sort of like glue. And it allows for unique immunoadjuvanted immune responses. And it's only there for a brief time. These are the crystals of microcrystalline tyrosine. And it was shown that MPL and microcrystalline tyrosine had synergy. That's that high bar showing allergen-specific immunoglobulin G dramatically increased versus MPL or allergen alone. This is a model allergen, ovalbumin. And he showed that it was more effective than alum, which is a common adjuvant, in terms of driving a Th1 response, which in an animal model is an immunoglobulin G2A response. And this is after a month. The big green bar in the center is the monophospholipid A tyrosine combination with microcrystalline tyrosine format, showing a bigger boost than you got on bar 6, which is with alum instead of microcrystalline tyrosine. So this was finally taken into the clinic using this approach about 2007. And I was fortunate to be the lead U.S. investigator on a study. We call it ultra-short specific immunotherapy, and we're successful in treating grass pollen. We had a number of luminaries who were co-authors: Friedrich Horak from Vienna, Anthony Frew, who was past president of the European Academy of Allergy, tragically passed away a few years ago, and some people from the company Allergy Therapeutics. So what we did was 4 pre-seasonal injections. The amount we gave cumulatively was 5,100 SU, and this SU is an internal standard developed by Allergy Therapeutics. And we did it in the grass pollen season of 2008. And what we showed was that there was a significant reduction in combined symptom medication score. In a complete data set group, it was over 20% benefit, 24% greater than placebo. And IgG rose. This is what we'd expect with successful immunotherapy. And there was no anaphylaxis. So it met all the criteria we want. We had 1,000 people in this study. This is a massive study. And it was international. It was United States. There are European sites, Austrian sites, and Canadian sites. Tony Frew later republished a different analysis of it, along with me and the other cohorts in the study, showing that if you looked at peak placebo scores rather than peak pollen counts, you got an even more dramatic effect. It was 26%. The idea being here, when placebo-treated people are sick, that's what you want to see. You want to see when they're at their worst, are you going to be better? And in fact, we were 26% better than they were. Peter Jan de Kam later came to Allergy Therapeutics as their research director. A lot of work was done to optimize this vaccine. I'll just go through it quickly. We found that our 5,100 dose was not sufficient. You needed to get up to 27,000. A dose-finding trial was done using conjunctival challenge. At 27,000, we had the greatest impact. We had a big boost in immunoglobulin G to the allergen, and we didn't see any serious adverse events. But this was with conjunctival challenge. We then did a field trial, and I'm one of the co-authors with a number of luminaries from Europe. And what we did was we gave the injections now, not 4, but 6 injections. We gave them either weekly or weekly for 3 weeks and then monthly for 3 months. And we found that the monthly for 3 months after weekly for 3 weeks worked a bit better. Our symptoms were the lowest in the green, which is what we call the extended protocol through the grass season. and everything was highly statistically significant in terms of the extended regimen shown on the top. All the endpoints that we looked at, combined symptom scores, medication scores, were all p less than 0.05. And we had a big improvement in terms of the combined symptom medication scores, which was the primary endpoint, and the improvement was greater than placebo with this extended protocol. Our IgG4 went up, our quality of life scores, our QLQ, improved greater with the extended protocol, and we had, again, no anaphylaxis. And that's really been the goal, to do this safely. Subsequently, we've done an international study that looked at this same approach called Resonate, and it's a phase 3 pivotal trial where we had an interim analysis after 1 year. It was supposed to be a 2-year study, but after 1 year we broke the data, but we had to have p not 0.05, but p of 0.014. So we had to have a lower p to show that it was effective, and after 1 year it was effective. So after breaking the data, it was stopped for success. These are the sorts of people. They had very high allergen-specific IgE, 28 kilounits to grass, and we showed a very nice improvement in terms of grass symptoms throughout the season. And in fact, the overall improvement that we were getting was in a range of 25 to 40%, depending on what parameters we looked at. Quality of life also improved. Allergen-specific IgG also went up dramatically. So all the things that are markers of success seem to be there. Mo Shamji, who's now president of European Academy of Allergy from Imperial, began working with us looking at biomarkers. And what we found is that with this approach, we got a big rise in allergen-specific immunoglobulin A1 and A2. Mo is now proposing as a criteria for successful immunotherapy, and a reduction in facilitated antigen binding to B cells, which also appears to be associated with success in immunotherapy. And we found that we had over a 90% adherence with 6 injections in this Resonate study. We did a meta-analysis of the 2 studies, G306 and G309, just to see where we were. And where we were was really similar to sublingual grass tablets and sublingual tablets for 5 grasses or 1 grass, Timothy grass, which are on a marketplace in Europe. And why that's important is those grass tablets, to have a long-term effect, you need to take them for 3 years, every day. Here, we believe we're going to get a long-term effect with just 6 injections in 1 year. So, I mean, it's a big difference. A long-term pediatric study has been begun, and we're hoping that we will have data showing that there's long-term benefit from the study, and I was actually in Poland for the investigator meeting in October. We'd love to bring it here, but because of the current situation, it's a challenge. I will say our next project after grass is going to be ragweed, and that's a big issue here. I was involved with the company ALK in the approval of their ragweed tablet, and we had study sites in Hungary, Romania, and in Ukraine. And that's part of what I was doing in Ukraine in the mid-2010s. So we think that when the ragweed begins, hopefully the conflict will have ended and we can be here. We're discussing collaborative projects. Well, this could be one with a modified ragweed immunoadjuvanted immunotherapy. Let's see, this thing is, well, I had a few more slides, but oh yeah, here we are. The next and final thing I'm going to talk about briefly is a peanut allergen vaccine we're developing. Now this is going to be the first ever, and this is maybe the most important thing I've ever done. And this is the virus-like particle. Peanut allergy affects about 2% of the population, and it can cause death. So unlike grass allergy, which usually doesn't cause death, this can actually be fatal. And what we've had up until now is oral immunotherapy. You got to swallow peanut powder every day. Not very practical. Omalizumab, very expensive. It will be available as a biosimilar in the US next year. It's already available in Europe. but you have to, again, use it every 2 to 4 weeks, and it costs on a order of $30,000 to $100,000 per year to treat with omalizumab. So what can we do instead? Well, this novel treatment, this peanut vaccine, was developed by Thomas Kundig and Martin Bachmann from Switzerland. And this is the virus-like particle. It's a nanoparticle. Attached to it in red is ARA-A2, which is the major peanut component allergen. And there's tetanus toxoid inside, on the outside, and there's E. coli encapsulated RNA inside. So there's 2 immune adjuvants, and the allergen is spread over this particle in such a way that you can't cross-link immunoglobulin E molecules. And in brief, clinical development was done beginning with animal studies. A lot of it occurred at Imperial in the UK with Mo Shamji, and they showed that given to laboratory animals, virus-like particle was not anaphylactic with this allergen on the particle. Animals that were sensitized to a model allergen didn't develop anaphylaxis. Skin prick test was reduced in these animals, and looking at a single allergen, we got a big immunization response. Mo Shamji later did some work looking at human subjects, showing basophil activation was reduced. The effect was large on dendritic cells, B regulatory cells, and IgE production. Here you see dendritic cell type 1 genes go up, type 2 go down. IL-10 in this case does go up, and T cell response shows interferon gamma Th1 response when you use this system, and reduced Th2 response. So reduced capacity to activate basophils, pro-tolerogenic dendritic cells, induction of interleukin-10 regulatory B cells, and reduced development of Th2 responses. So this study is called PROTECT, and it's ongoing now. First, healthy subjects was studied and was found to be safe. None of the healthy subjects developed any problem in terms of testing with it. More recently, peanut allergic subjects have been tested with it. It's been found to be safe. We're still doing dose ranging of that with 4 ascending doses, but we think we can desensitize someone with just 3 injections and get potentially a long-term benefit using this system. So far, our number of subjects are small, so we're under 20 people that have been treated, but it's been safe, it's been well tolerated, no systemic reactions, and in peanut allergic subjects, what you see is a big boost in RA2 IgG, and you see a major improvement in terms of skin test response. I don't know, it looks like I lost this again. Yeah, skin test response goes down. Wayne Schreffler from Harvard just presented at the American Academy of Allergy last month evidence that IgG goes up, IgE associated responses goes down, basophil activation goes down after treatment. So key messages from all this is that virus-like particle may be a system that could be applied to any food. And if this works, the potential for using any food on this virus-like particle and getting an effective desensitization. And so far, so good in terms of immune response and tolerance of it. Low doses are only needed because of the way the particle works, and these are all the collaborative sites that are involved. So this is our team. Here I am with Peter Yandakam, Mo Shamji, and Matthias Kramer from Bencard, and Janice Lahady from Imperial. And again, it's the most exciting thing I've ever been involved with. So in conclusion, modified allergens are a safe way to give immunotherapy, but you need an adjuvant. Ragweed may be next. We hope we can come here, and we hope it will be possible to do so. And I want to give a quick thank you, and Slava Ukraini.
Thank you, Professor Lawrence, for this beautiful lecture for us. Do you have any questions for Professor? Please.
Thank you, Professor, for your lecture. Based on the sheer knowledge, I'm at the absolute bottom in this lecture hall with all my professors and doctors here. My question is just from my curiosity. So this adjuvant system, it only works on type 1 hypersensitivity, or it can also be used for type 2, 3, and 4?
Well, in this case, we developed a system that specifically modulates from type 2 to type 1. So I mean, this is very specific to get in one direction. And the reason is allergic responses are type 2 driven. We know that. And this adjuvant that we chose, monophospholipid A, micropristinal tyrosine, saponins in the case of the zoster, this was designed to give us a big T1 response. Now, in the same vein, with immunization, you want to get a T1 response. That's why the adjuvant works when you want to immunize for a virus. The adjuvant works when you want to convert an allergic response to a non-allergic response. Because in both cases, we're driving in a T1 direction. But I think this is the way medicine's going to go, quite frankly. The world has changed dramatically over the last 50 years. 50 years ago, I was treating asthmatics with drugs like theophylline. We were treating people with drugs that were affecting some of the, what I would call the structural aspects of disease, but not affecting what was actually causing disease. And how the world has changed, whether you're talking oncology, rheumatology, allergy immunology, infectious disease, is to get at the immune mechanisms. And that's why 50 years after getting in this field, I'm proud to be an immunologist, because I think the immune system underlies most of what is happening in our body. We used to say we're the only system that doesn't really have a specific organ, unless you want to consider the lymph node, but where everybody else is organ focused. Here we have a situation where you're thinking about an infectious disease, you're thinking about a malignancy, you're thinking about allergic disease, wherein you can use what is specific to that disease, like an infectious disease with RSV and zoster, like the case of an allergen, or like the case of a tumor. You take a tumor marker, combine it in an adjuvant system, and get the body to change, and change in a profound way that can save your life. I mean, we're looking now at adjuvant systems, for instance, for pancreatic cancer, that people are now living. We're looking at bladder cancer that used to be a death sentence, and now people are living with adjuvanted immunotherapy as the technique. So, we've gone from the simplest ways of approaching disease, irradiated, bronchodilators, antihistamines, to ways of addressing what is actually causing the disease and changing the way that the immune system, the body, is responding in a way to get us modulated away from the disease. I think the next step after all of this, and will be the most interesting in the end, will be genetic therapies. And that's where this is. Because once we understand the mechanisms, and we show that we can induce a mechanism that drives you away from disease, next thing you know, well, can we do that on a gene basis? And my prediction is we will. And this will have a dramatic change in how we practice medicine. So anyway, thank you very much. Great question.
Thank you.
Any other questions? Thank you, Professor DuBuske, for your perfect lectures, for your visit to our university, for your great support of Ukraine. You told about this support, and now we know that your influence to some congressmen was very important to to change the opinion about support to Ukraine. And from all Ukrainians, from all our university community, we tell thank you, and we applause for this. I think that your visit to our Ukraine, to our university, will start new cooperation between our university, between your organization, and nowadays I want to tell that we have a very big European grant, Horizon Europe, Clean Air. We investigate the influence of pollution of allergic rhinitis, and one of our friends, our partners, is European Academy of Allergy and Clinical Immunology, and we well know Maria Torres. We met her in different meetings, and I think we can establish any other new cooperation between Ukrainian and American institutions. And I want to award you with this certificate from Bukovinian State Medical University for Lawrence DuBasque as the clinical professor of medicine at the George Washington University School of Medicine and Health Science, Director of Immunology Research Institute of New England, for outstanding personal contribution to development of American-Ukrainian relation and support of Ukraine, promotion of educational, moral and scientific values among generation of students, as well as fruitful cooperation between our institution. I want to invite with this certificate you. And thank you all for teachers, students, all who participate in this meeting, this lecture. Thank you. Good luck. Slava Ukraini. And we will make photo together with Professor Lawrence DuBasque.
And I'm serious, I hope that we're back here with this project when it comes to that. Yes. A big issue here with the